The overall goal of this procedure is to detect fluorescent nanoparticle interactions with primary immune cell subpopulations by flow cytometry. This is accomplished by first treating the cells of interest with the nanoparticles. In the next step, the cells are labeled with antibody against cell-specific surface markers, and then analyzed by flow cytometry. Ultimately, the interaction of the nanoparticles with individual immune cell populations can be measured.
The main advantage of this technique over existing methods like microscopy is that with this technique variations in fluorescence intensities induced by nanoparticles can be quantified in non-adherent cell populations.
For nanoparticle internalization in blood leukocytes or purified monocytes, begin by plating 5 × 10⁵ of the cells of interest in 1 ml per well, in each well of a 12-well plate. Then add 10 microliters of freshly prepared working fluorescent silicon dioxide nanoparticle suspension to each experimental well, adding an equal volume of complete medium to the untreated control wells at this time as well. After a 1-hour internalization at 37 degrees Celsius, transfer the samples into individual 1.5 milliliter polypropylene tubes, and then spin down the samples for 3 minutes at 6,000 times G and room temperature. To stain the cells with CD14 VioBlue, incubate 100 microliters of each cell suspension in 10 microliters of the human antibody at a 1:11 ratio.